I am Chengxin Zhang, an HHMI postdoc at the Pile Lab at Yale. Today I introduce rMSA, an automated pipeline to generate multiple sequence alignments, or MSA, for a query RNA sequence. MSA is the entry point for many applications in RNA structure modeling tasks. For example, co-evolving patterns across different positions can be computed from the MSA, where pairs of nucleotides with more commutation events are more likely to physically interact. This information can be used to inform canonical base pairing and tertiary contacts, which can be further used to guide RNA 3D structure prediction. Many methods developed for MSA-based RNA modeling are trained on MSAs from the rFAM database. This can be problematic because of the exceptionally high quality of rFAM MSAs. Many rFAM families are for relatively well-characterized RNAs with lots of homologs. 5' and 3' ends that are not well aligned are excluded from the MSA. More importantly, the initial alignments are manually curated. These semi-manual curation processes make it very difficult, if not impossible, to generate MSAs for less studied RNAs with alignment quality as high as those from rFAM. If an MSA-based RNA structure prediction program needs to be applied to RNAs outside rFAM, it must be trained on automatically generated MSAs. Unfortunately, there are very few programs that can automatically perform database search and alignment for a target RNA sequence. BLAST-N performs sequence alignment, while NHAMMER performs HMM sequence alignment. Inferno uses a special format of sequence profile called covariance model and performs covariance model sequence alignment. Inferno requires an initial MSA and secondary structure to construct a covariance model. rNA-CMAP automates Inferno by using BLAST-N alignments for covariance model construction. In this work, we develop a more sophisticated approach for MSA generation. Our RNA multiple sequence alignment, or rMSA protocol, has 5 stages. In each stage, an Inferno format covariance model constructed with different parameters is searched against the RNA Central or NCBI-NT database in an iterative fashion. One final MSA is selected from the 5 MSAs by the number of effective sequence or by covariance scores. We compare rMSA against 4 other existing MSA programs by testing their MSAs for prediction of RNA secondary structure using 2 different covariance programs, PLMC and RScape. The accuracy is measured by F1 score for predicting canonical base pairs. For both PLMC and RScape, rMSA significantly outperformed the best third-party program, rNA-CMAP, by around 20%. We also tested the MSAs for predicting contacts using a deep learning program, rNA-Contact. Here, a contact refers to any nucleotide pairs within A angstrom. Again, rMSA significantly outperforms all other MSA generation approaches by at least 4.6%. We further apply rMSA for long non-coding RNAs or lincRNAs. Many lincRNAs are known to have stable secondary structure. However, whether these secondary structures are evolutionarily conserved has become a topic of recent debates. For example, a 2017 study found that the covalence program rScape cannot find any significant base pairs for at least 3 linked RNAs, including hot air, SRA, and the rAA region of cyst. This conclusion is reiterated in 2 subsequent papers in 2020 and 2021. This analysis, however, is inconsistent in terms of MSAs. In particular, rScape was originally trained on high-quality RFM MSAs, but when tested on linked RNAs, this study only used shallow and low-quality MSAs collected from various experimental literatures. To track the impact of MSAs on this analysis, we repeat the same rScape analysis on the aforementioned 3 linked RNAs. On the right-hand side table, reverse MSAs are the MSAs from the previous 2017 study, while rMSA is our MSAs. Under the same default rScape setup, no base pair can be predicted from reverse MSAs. On the other hand, from our MSA, rScape predicts 30 and 4 significantly conserved base pairs that overlaps with experimental secondary structure of hot air and rAA respectively. In the previous study, the 4 domains from hot air are studied separately, but rAA and SRA are studied in full length. If we also partition rAA and SRA into domains and run rMSA on individual domains, rScape can predict even more base pairs. For example, 0 base pair can be predicted from the MSA built for the full-length rAA, while 46 base pairs can be correctly predicted from the rMSA alignment built for rAA domain 1. These results show the strong impact of MSAs on covariance analysis. In conclusion, we developed rMSA, an automated algorithm for RNA MSA generation with higher quality than existing programs. rMSA alignments and domain partition allow detection of evolutionarily conserved base pairs in linked RNAs previously considered to lack conserved secondary structure. With that, I would like to thank colleagues in the Power Lab and the Zhang Lab. Thank you for listening.